Good, recording in the room. Welcome back, everyone, to the Water, Wetlands, and Watersheds Seminar. It's fall 2021. It's still 2021. It's Wednesday, October 20th. And today we are very pleased and lucky and thankful to have Dr. Cynthia Heil speaking with us. And so let me find the right screen here. So Dr. Heil is a senior scientist at Mote Marine Laboratory, and she's the director of Mote's Red Tide Institute, which is focused on developing and testing new technologies for the mitigation of Florida red tide. And we're gonna hear a lot about that today. Dr. Heil first developed her interest in marine science and red tides growing up on an island off the Maine coast. I don't know if we'll see any pictures of that, but certainly red tide that has occurred up in that part of the world as well. She has her BS in cell biology from Purdue University, MS from USF's College of Marine Science, and her PhD from the Rhode Island Graduate School of Oceanography. She did postdoc in Australia and at USF and has worked on developing methods to assess water quality and the physical, chemical, and biological factors that are responsible for red tides. And I think we'll hear a lot about that today. And additionally, methods really targeting the mitigation of existing red tide and hopefully the prevention of future red tide. So I'd also say she was the leader from 2003 to 2010 of the red tide group at Florida's Fish and Wildlife Research Institute, which I'm sure inspired a lot of the work you're doing now. So without further ado, Dr. Heil, the floor is yours. You can go ahead and share screen.
Okay. How's that? Can you guys see it?
Nope.
No. How's that?
So you got to go to the green button at the bottom and
Oh, I thought I hit it. Wait a minute.
share screen and then select that window with the Google Doc in it.
All right. And share screen one. How about that?
That's perfect. We see it and it's looking great.
This is why I have 25-year-old technicians. Thank you, David.
You did it.
Yeah, finally. Welcome, everybody. And I thank you for taking your lunch out to hear a little bit about red tide and a current mitigation research we're conducting here at Mote in collaboration with a lot of other scientists around the state and the country. I always like to, let's see, wait a minute, start with a roadmap of where I'm going to go in this talk. A little bit of background, a little bit on HABs in general, Florida red tide. Florida's rather checkered red tide mitigation history, bombing dead fish and flamethrowers, believe it or not. How we currently test compounds for mitigation here at Mote, and what is the future, the Florida's Red Tide Mitigation and New Technology Initiative. So at the very beginning, what are phytoplankton? I'm hoping most of you know what phytoplankton are. They're probably the most underappreciated primary producers in the world. These are the microscopic, single-celled, mostly photosynthetic aquatic plants that you're probably never even aware of. They are, yeah, you owe your existence to phytoplankton. They're the base of the food chains, responsible for 2/3 of the world's photosynthesis. Probably about 75% of the oxygen you're breathing came from a phytoplankton cell. And in current times, they're crucial for climate change. They sequester carbon dioxide, transfer it to the ocean sediments. Probably the reason why we're not 3 or 4 degrees warmer than we should be right now. Now, that's the good. There's the bad. Some of the more than anywhere from 10,000 to 100,000 species of phytoplankton are either harmful, we call them harmful algal blooms, or HABs, and this is about 300 species. And some are toxic, about 100 species. And these are toxins that are some of the most toxic known to man reside within phytoplankton. And one of the reasons it's a problem is these blooms, these HABs, are increasing in frequency, magnitude, duration, and toxicity. Not every HAB, but a lot of them are. Here in Florida, we have a lot of HABs. The state currently monitors for more than 75 species of HABs. 3 of them are regulatory, Karenia brevis, Pyridinium bahamense, and Pseudonychia. 1 is non-regulatory, but it's arguably the greatest influence globally. And we have a whole variety of HABs of what we call concern. Cyanobacterial HABs, green slime, red drift algae, macroalgae, and EDABs, ecosystem disruptive algal blooms. And these are essentially non-toxic blooms that become so dense they disrupt the ecosystem. We are concerned with one right now, Karenia brevis, and these are the ones that impact probably the most amount of people in Florida. A little bit on the background, you probably all know, so I'll briefly review this. Karenia brevis blooms, and it's a non-thecate dinoflagellate. In the upper right, there's a light micrograph and a scanning electron micrograph. They're native to the Gulf. They bloom annually in the Eastern Gulf. We have over 13 species, but Karenia brevis is the one that concerns us the most. There are human health impacts, short-term and long-term. Notice the question marks on the long-term. We know next to nothing about these. There are environmental impacts. Probably the best example I can give you is this past summer in Tampa Bay, Pinellas County removed over 1,400 tons of dead fish that had been killed by red tide. So they can have huge environmental impacts and some more recent economic impacts. Losses estimated to be up to $1 billion for the 2018 bloom. And it's a whole variety of sectors we're talking about. Why is Florida so prone to red tide? This is 65 years of data from the long-term database, and it's essentially grouped into the number of years in each of those regions with red tide. So Southwest Florida has a lot of red tide, probably the most in the Gulf of Mexico. Why? This particular species, a few things you need to know about it. It's motile, it swims at about 1 foot an hour, and it's a very poor competitor. It grows very slowly. It is a K species. And it's always been a puzzle how it blooms because it is such a poor competitor. And what we think is a lot of it has to do with the bathymetry of the Gulf of Mexico. You hear about the West Florida shelf, and this is a map that kind of shows that very, very graphically. We know that blooms start offshore on this West Florida shelf. It's very shallow, out very far, oligotrophic, not a lot of nutrients, not a lot of competitors. And the loop current comes in, which is this current that moves up through the Yucatan and then down through the Florida Straits and rejoins the Gulf. Periodically, it impinges on the shelf. And what this does is it brings nutrients with it, and it also essentially starts to push a bloom near shore. So as this bloom is slowly growing, the currents are slowly moving it near shore over this wide shelf where it doesn't have a lot of competitors. So by the time it hits near shore, we have a very concentrated bloom. And this is how these blooms start. And there are a whole variety of nutrient sources I won't go into near shore that support this bloom. Over 13 different nutrient sources, that's another talk. So once it gets near shore, the blooms are usually pretty happy. So this is kind of the heart of the talk. What can we do about red tide? Is HAB science sufficiently advanced these days to think about mitigating blooms again? And I'll get into the again in a minute. But I need to explain a little bit about mitigation. When you tell people you're researching red tide mitigation, their first gut instinct is, oh no, radioactive clay. You're dumping clay into the water. Mitigation is a lot more than just putting clay into the water. There are 3 mitigation strategies: prevention strategies, control strategies, and actual mitigation strategies. And they all fall under this big umbrella of mitigation. Prevention, these are the hardest to do, but the most preferred. Stopping the bloom from occurring in the first place or limiting their extent. You can see why this is hard to do. These blooms start offshore, anywhere from Tarpon Springs down to Naples. So it's about once every 3 or 4 years that we actually find initiation. It's sort of a hit or miss at this moment. Control strategies, which focus on limiting the impacts of the bloom by either killing the cells or removing the cells and the toxins from the water. This is where a lot of the current efforts are. And mitigation strategies. Essentially, you're not so much working with the bloom as you're trying to limit the impacts of the blooms. You're not dealing with the cells directly. This is probably the easiest thing to do. Monitoring, harvesting bans, removing dead fish from beaches, satellite remote sensing, forecasting models. So a lot of the prior 30 years have focused on the third mitigation strategy here. Right now, the state programs and the research we're doing is kind of a mix, mostly control strategies, but some mitigation strategies we'll get into. First rule of red tide mitigation: do no more harm than the bloom itself is doing. We don't want to make the situation worse. Anything we put into water cannot kill other things and cannot make the bloom worse. At the start, you remember saying, are we advanced enough to deal with red tide or research it again? Florida has a very long, very checkered history of mitigation research with regards to red tide. We know red tide has been around since the 1800s based on neurotoxic shellfish poisoning. There used to be oyster houses in Tampa Bay, and occasionally people would get very sick eating oysters. The symptoms were very characteristic of brevetoxin poisoning. They're very unique, so we know that what was happening. Respiratory irritation, first reported in 1912. And you think about it, that's when people started going to the beaches here for the first time. But it really wasn't until 1947, during probably the worst bloom on record, that Karenia brevis was identified as the cause of these blooms. It was described taxonomically and named. And the first mitigation attempt, or suggestion, I should say, was in 1947. And that little red circle and that blank should be a front page of the the Sarasota Herald from 1947. It didn't transfer to the talk. But Senator Claude Pepper, who, if you know Florida history, you will have heard the name, was so aghast at this red tide that was literally knee-deep in dead fish on the beaches that he requested the Rear Admiral in Pensacola to send his bombing planes down to literally bomb the fish off the beaches. And the Rear Admiral luckily was very sensible and replied that it was inadvisable, as it's likely to disperse the present concentrations and involve additional beach communities, which it definitely would have. Senator Pepper went on to suggest then that we use flamethrowers to eliminate the dead fish from the beaches, which is kind of a variant on the dead fish are now removed. In some counties, actually incinerate them. So it's always interesting to know your history. But probably the most relevant here was the early studies in the 50s on mitigation. So these were just suggestions initially, nobody took them up, but in the early 50s, Florida, in collaboration with federal agencies, investigated copper sulfate. And that's a compound that's used, still used in a lot of freshwater HAB mitigation. But they went right to the source of bloom off St. Pete and literally dumped over 100 tons of copper sulfate into this bloom over a period of time, literally from Clearwater down to St. Pete Beach. Applied it with burlap sacks between the bags. And when they couldn't get enough copper in that way, they brought in crop dusting planes and literally crop dusted 100 tons of copper sulfate. And keep in mind, this is the 1950s. Nowadays, it's kind of horrifying to actually read of this, but it was a lesson well learned. And basically, yes, the copper killed the cells, but when you lyse cells, the brevetoxins don't necessarily go away, they just dissolve into the water. So any mitigation has to deal with cells and you have to deal with the toxins. So essentially while they killed the cells, they released the toxins and made the bloom more toxic. And then 2 weeks later, the tides brought the red tide back in. So that was kind of a lesson well learned, as you can see from the quote I put there, at least in Florida, not necessarily in Texas. They also did an early study. They do get red tides in Texas of Karenia, just not as frequently as we do in South Florida. They tested, in this case, copper ore on a lagoon connected to the shipping channel. And it was interesting because they got completely different results. They added 20 tons of copper ore. A hurricane came by and they added another 40 tons 2 months later because they concluded that the hurricane actually disseminated the first effort. And the interesting thing is they didn't see any toxic effects. There were no toxic or harmful effects. A lot of speculation as to why, possibly chemical interactions with chelators, suspended material, but essentially there was no impact on the phytoplankton communities. So you're seeing the do no harm and then a theme of the Florida studies. And then with Texas, it's more, we're dealing with an incredibly complex environment here that you are manipulating and you have no idea what the consequences are going to be. So there needs to be a lot of preliminary research. And there was some research done, oops, I'm getting ahead of myself, in the lab in the 60s. One study screened almost 4,500 compounds. And it was interesting that the majority were non-toxic against Karenia. So almost 200 were toxic at higher concentrations. Only 5 killed Karenia brevis cells after 24 hours at lower concentrations. And of those, only 1 consistently killed Karenia. And when they tested that compound with other organisms, it was very good at killing other organisms too. So this essentially is why there's probably a 50-year gap in our mitigation research with red tides. These were lessons learned early and learned hard. So when you think about synthesizing all this, what does it tell us about mitigation? It's not simply adding clay to the water to get rid of red tide. You have to look at effectiveness on Karenia cells, on the toxins, and on the environment. You have to look for side effects. Are we changing the habitat? Are we impacting other species? Are we impacting humans with this? 100 tons of copper sulfate went somewhere in that system in the 50s. We have no idea where. And there are certain societal questions too. Do we want to mitigate not very severe red tide? Does it have to be severe? What are the practicality, the logistics and costs, and the actual laws? So if you synthesize all this for an ideal mitigation technology, these are the three conclusions. It's got to be ecologically sound, economically feasible, and logistically attainable. And that is a lot to ask of anything. And this is more of a graphic representation of what I'm talking about. You need to control the bloom, and there's cells there, and that's actually the brevetoxin structure. When you look at a bloom, there are cells, but there are also dissolved brevetoxins present. So you want to eliminate the cells, and you want to eliminate the cells so that you don't produce more brevetoxins from those lysing cells. So whatever compound you're looking at, it has to work on cells and it has to work on toxins. Or you want to control the impacts of the bloom, reduce aerosol production, reduce the environmental impacts, such as removing dead fish, or alter human behavior. We know there's a bloom, don't go to the beach. And that's a question of educating people and monitoring. So where do we start on the research for this? I started with Mote about 2.5 years ago. When I arrived, it was right after the 2018 bloom, which was almost equivalent to the 47 blooms in terms of severity. It was bad. Scientists here at Mote had started collaborating with certain companies and scientists with state funding on testing mitigation, and they developed a tiered testing system that I'll briefly describe too. So what did I do? Well, I know that in the Southeast Asia, there's a huge HAB problem, and they've been working on mitigation of these HABs for the last 30 years. So as a scientist, the first thing you do, you go into the literature. And what we did was do an extensive troll, found about 125 potential compounds that we thought might be effective. So we pulled those and screened them for a variety of things. Do we think they'd be applicable to a non-thecate dinoflagellate? What the cost? If it was $500 for 5 grams, then obviously it's not going to work. Availability. There's some very effective herbs grown in Western China that you can't get your hands on. Practicality. A few compounds you can only buy a ton of on Alibaba. So that was out for testing. We settled on 25 resulting compounds that we've been testing in the lab. And also about that time, the new red tide mitigation and new technology initiative started. And this is a focused state effort on mitigating red tides that I'll go into in a minute. How do we test compounds? I mentioned the tier testing system. This is a scientific process. We have a compound. We essentially start at tier 1 with lab experiments. We want to test efficacy. Does it work on cells? Does it work on toxins? If it does, then we need to know what the most efficient dose is and the application method. And I'll show you examples of why this is difficult in a few slides. If it passes, if it doesn't pass the efficacy or we can't figure out the dose, then as Dr. Crosby here says, it goes on to the off-ramp and we move on to the next compound. If it is effective, though, we move it up to tier 2 mesocosm testings. And this is the new mitigation mesocosm testing facility here at Mote. We try to work with natural communities or cultures, supplemented with cultures in much larger volumes. And we're looking at toxicity tests, adverse impacts, logistical issues. How do we scale up something that works? Are we seeing negative effects on other organisms? And if it passes that, then we move it to pilot studies in canals, in floating mesocosms, floating limnocorals, and I should say with appropriate state and federal permits. And that's about a year-long process to get these permits right now to put anything in the water. So what worked and what didn't? I am going with the stoplight, green light, red light. And this, just to explain briefly, this is kind of a summary, and I see the bottom of the slide didn't work out very well. Our experiments we run for 48 hours. We test cells and toxins both at 24 and 48 hours. So the compounds are listed on the left or the chemicals. And what it did was to cells in 24 and 48 hours. If it's red, didn't work. If it's green, killed more than 50%. And the same with brevetoxins to the right. And what you can see is there's a whole lot of red in the brevetoxins, a lot more green in the cells. So there were a lot of things that worked on cells that did not work on toxins, essentially. It's much rarer to get something that worked on both. Some compounds took the whole 48 hours to work. Some compounds were immediate, especially with some of the companies. So there's a whole variety of impacts. But as you can see, we need to test the cells. And what a lot of the literature doesn't do is test the effects on toxins. And that's something to be aware of. So there are some promising things in here. And I just wanted to briefly review maybe 3 or 4 of them that we've proceeded with from the initial screening. And I should, the three at the bottom, we were testing actually not the effects so much on cells, but the effects on aerosolization. Do these compounds modify the aerosolization of brevetoxins? And I'll get into that in a minute. And the answer is actually yes for the toxins, no for the cells. What didn't work? And I put this slide up here for a reason. I mentioned things work on the cells and not on the toxins. Another category is some things actually do work on the toxins, but what they end up doing is converting them into another form. In this case, we're looking at chitosan with additions of modified clay, which has been used extensively in Vietnam for some of the shrimp farms. And this is essentially shellfish skeletons, dried, ground up, chitosan, they're sugars. The lower left is what happens to cells when we add it alone and with sand. And essentially, the black line is the control, and really nothing there is different from the control. Didn't have an effect on the cells whatsoever. Now, if you look at toxins on the right, this is a % decrease in toxins. So initially, and here's PBTX1, which is brevetoxin 1, brevetoxin 2, and brevetoxin 3, or 3 different forms of brevetoxin. You can see it was within 24 hours, it was fairly effective with brevetoxin 1 and 2. Didn't seem to do anything with brevetoxin 3, but after 48 hours, what we see is a negative % decrease. So we're seeing an increase in brevetoxin 3. So this particular compound, yes, was effective on 1 and 2, but it was converting them after 48 hours to brevetoxin 3, which is more toxic. So essentially the compound was making it more toxic. So lesson learned, you do need to measure toxins, and you need to measure the forms of the toxins. So this one got the off-ramp. We have been working with seaweeds, ulva and gracilaria, and this is in collaboration with a lot of other scientists here at Mote. There is an extensive literature on allelopathy, which is essentially chemical warfare between plants. They put out compounds that deter the growth of other plants. And we sent the interns out one summer and said, bring us back any type of seaweeds you can find. And we tested those at the bottom. And these are simple % reduction in cells and % reduction in total brevetoxins. And you can see gracilaria and ulva were fairly good at reducing cells, did have an impact on toxins too. Other species, not so much. One actually increased the red tide, and 2 other ones were less effective. So we focused on gracilaria and ulva primarily. And these initial experiments were a lesson in dosage and dose form, because we have ulva growing behind our lab like weeds. So we took it and dried it, ground it up, added it to red tide, and it was very good at killing cells and toxins. The problem we found out is when you do that, you introduce a lot of nutrients back into the system, which is essentially a nutrient source to grow more red tide. So it's a little counterproductive. So we moved away from the dried, and we've been testing either living ulva, putting, as you can see, the blades of the seaweed in, or what we call ulva tea, which is the media where the ulva has been growing. And we're working toward using PUFAs, extracted PUFAs, polyunsaturated fatty acids, which we think are the compounds responsible for the effects we're seeing. So macroalgae is a lesson, and it's effective, it's attractive because it's natural, it's there already. However, we have an issue with the form in which we apply it and the concentrations that we're slowly working out. Believe it or not, curcumin, those of you who are interested in your heart health will know it, turmeric. We've been testing that. That was a compound also used in China that has worked very well. It has human benefits. It's an antioxidant, anti-inflammatory, but it also works on harmful algae. And as you can see in the lower right, % reduction from control. It's 100% of cells at moderate concentrations in 24 hours. It's very effective at cells, and it's pretty good at toxins too. Up to 70% reduction in toxins, which is fairly good for what we've been seeing. Our problem with this right now, and we think we have a workaround, we're investigating it further, but curcumin is turmeric. If you cook with it, you know it's orange. It's very orange. So any beaker we've added it to, it's bright orange. And it's been pointed out we can't really add it to a bloom and turn Sarasota Bay orange. So we've been working with actual growers of turmeric in Hawaii, and they have a clone that is non-colored. So, or they've extracted the color from it. So we're hopeful, and we're pushing this. We've completed tier 1 testing where we're moving up to mesocosms with this. Quats is another thing we've been working on. This is short for quaternary amine compounds. These are FDA-approved surfactants. Basically, they bind to negatively charged cells and disrupt the membranes and disrupt the enzymes. And why they're so attractive to use for HAB mitigation, they've been used for cyanobacterial mitigation, is because you can adhere them to a substrate. So you can adhere them, in this case, to concrete meatballs or fiberglass tape, which is what we've been doing. Put them in the watertight and then remove them again when you're all done. So rather than clay or rather than curcumin, where you put in the water and you cannot retrieve, with a quat, you can put it in and you can take it out again. They have proven, I won't say surprisingly, but very effective against both cells and toxins. There's a graph showing the blue controls on the right and the treatments, and the toxins on the bottom, and show it reduces all the forms, 1, 2, and 3, brevetoxin, as well as the congeners and brevinol too. Right now we're moving up to tier 2. We've decided that the fiberglass tape seems to me the most effective. We adhere the compounds with a polymer to the fiberglass tape. We're just wrapping our head around the logistics of how we do this with a 5,000 liter tank. Do we get sheets of fiberglass and put them in, or do we construct some sort of apparatus to do it? But it's proven promising, and we're following that up right now. Clay flocculation, I mentioned briefly. I'm not directly involved with this, but I just wanted to give you a slide on what it is, because it is ongoing research here. Mote scientists are conducting in collaboration with Fish and Wildlife and Woods Hole at the moment. And the idea here is basically you add a clay slurry at very low concentrations, and the particles collide with the cells and adhere to them. And they also adhere to toxins and sink them down to the bottom. We do not use radioactive clay from Mosaic. We actually are collaborating with some Chinese collaborators who have developed a specialized kaolinite clay that is used for this. And there were, during the summer, we did have some permits in place, so they were doing tests with these off the docks here in Sarasota Bay. And one of the last control mechanisms I wanted to talk about was ozonation, because this was developed here at Mote quite a while back. If you've been here, you know we have an aquarium with manatees, otters, sharks, rays, fish, all sorts of things. We take the water intake to maintain these animals directly from Sarasota Bay. So if there's a red tide here, and right now there's a red tide here, it comes in on the water and it affects our animals. So what the aquarium facility developed was an ozonation system, essentially applying ozone. And this kills the cells. They found it degrades the toxin, and at the same time actually reoxygenates the water. And there's an example, New Pass water in the bottom with red tide, and then after 12 hours in the lower right when it's treated. So it's quite effective against red tide. And you think, well, this would be, why aren't we using this right now? Well, Dr. Pierce's lab here did do a study in collaboration with Boca Grande community in a back canal a year or two ago, and brought the ozonation machine down and essentially used it and on the bloom in the water, monitor the water quality. It was quite successful, but if you actually look at the third slide to the right on the top, you can probably see the limiting factor with this particular system at the moment. It's the engineering. It sits on a dock and the water gets pumped through it. So realistically, you can probably make a dent in a canal, but a larger volume of water, it's at this stage where the engineering needs to be further engineered, essentially. So we have a system that works, but we need to engineer it for a larger volume or something in situ. I mentioned briefly that mitigation is not just control, it's actually mitigating the impacts. And one of the things we're looking at is, can we reduce the production of aerosols? And one of the ways we're looking at that is with surfactants or biosurfactants. In this case, we've done experiments with humic acids or tannins extracted from the Peace River. And these are dissolved organic carbon compounds that color the water brown. We extract those and essentially add it to a red tide. It modifies essentially the surface tension of the water, and it reduces the aerosol production, which is those little droplets that contain the toxin. And we've shown it can be quite effective at reducing aerosolization. So we're further testing a variety of different compounds and trying to wrap our heads around the engineering of this, how it will work. Do we apply it in the ocean next to a waterfront seafood restaurant? There's various suggestions localized at a beach. These compounds are naturally present. We're just concentrating them essentially and looking at the impacts. They don't affect the cells themselves, but they do affect the production of aerosols. And another project we're finishing up with Dr. Mike Parsons at Florida Gulf Coast University is how do we mitigate dead fish? In the simplest form, you pay somebody to haul the dead fish away, which is what happened this summer in Tampa Bay. As I said, they removed 1,400 tons, Pinellas County, in July alone from the red tide in Tampa Bay. There's a variety of reasons to do this. Dead fish are the #1 nutrient source for blooms, believe it or not, of the 14. If you have dead fish, it is a greater source than stormwater runoff, believe it or not. And the question is, if we remove those fish, are we removing a nutrient source? We're also doing a cost-benefit analysis. Each county removes fish in their own way. Some take it away and incinerate it. Pinellas County, until recently, Sarasota County didn't do anything with the dead fish. Economists are looking at, okay, what's the most effective way to mitigate these fish for the counties? How do they remove them? How do they get the biggest bang for their buck? And then the third part of this project, we're actually evaluating if we can compost these fish. Most times the fish are hauled away either to the dump or incinerated. They used to be buried on the beach, and hopefully they don't do that anymore. It's still a toxin vector when it's on the beach. But we've teamed with a company that makes a compost accelerator and testing whether we can compost that fish fast enough to produce a fertilizer that is utilizable. And the basic question is how fast do those brevetoxins in the fish decay? Luckily, I still have technicians after this summer. We retrieved 500 pounds of dead fish from Clearwater Beach in August, and it was an interesting trip with the swarms of dead flies following us over the Skyway Bridge. Luckily, they're doing the composting down at Florida Gulf Coast University, and we're waiting for the final data at the moment on the composting to see how fast the brevetoxins decay. But if it's fast, then this may be something that we can actually use those dead fish to produce a product. So the way forward, and I was going to conclude with this, I'd mentioned at the start the Florida Red Tide Control and New Technology Initiative. This is there's also a NOAA program that I won't go into, but this is a state program started in 2019. It's a 6-year dedicated program. Governor DeSantis signed it into law in 2019, and it essentially allocates $18 million over these 6 years to focus on mitigation technologies and approaches to address impacts of red tide. It has multiple parts, infrastructure in terms, if you want to assess mitigation, you need access to cultures and you need access to places to do it. So we've developed facilities here that do that. There are a whole variety of projects currently underway. I'll list a few in a minute. Some Mote-led, some partner-led. There's a TAC, a technical advisory committee, meets biannually and reviews it. We're leveraging a whole variety of private and federal funding for this. And we do report to the legislature. And I've listed the 2001 accomplishments priorities report at the bottom. If you're at all curious, I encourage you to go take a look. This program has funded, we currently have I think about 25 projects underway or just completed. And I've listed some of them. They run the gamut from flavonoids derived from beer products, believe it or not, are detrimental to HABs, to shrouds with integrated algicides, new detection technologies, new technologies to actually apply, monitor blooms and mitigate, apply the compounds, and then some companies with their specialized technologies, UVC LED technology, nanotechnology, which are magnetic iron particles with a nanotechnology cloud around them. We're testing. So this program is currently underway. If you're at all interested or you have an idea for mitigation, I encourage you. Every year there's a request for proposals, usually around December. These are one-year projects, anywhere from $50,000 to $250,000. It's a very simple 5-page proposal. And I'd encourage you to send an email to Kevin Claridge if you're at all interested. They are funding everybody from private citizens to companies with potential projects to academic scientists. So it's really an interesting program that's trying to pull in all the best ideas out there. And this request for proposals is annually. So I'm sending a invitation for everybody if you have an idea to apply. So what am I going to conclude from this? Let me see how I'm doing on time. Don't have a clock.
Doing great, yeah.
Doing good? Okay, maybe some room for questions at the end. There's really no magic bullet cure for red tide. I know it's an interesting thing about red tide research. It's a very complex phenomena. It's a complex organism. Its biology, ecology is complex, but people want to point a finger, and they want a simple solution. And the problem is there really isn't one. And just like studying red tide, mitigation is the same way. There's probably no one magic bullet cure for red tide in terms of some compound or some technology to control it. Each of the technologies, each of the compounds we look at has advantages, disadvantages, and also different potential uses. But we're hoping that through the rigorous scientific testing that we're learning something about all three of these for each of these techniques. So we're developing kind of a toolbox that we can call on, saying, okay, if this is our situation, a dead canal, nothing's living, we can apply this technology. But if we want to do something the whole Sarasota Bay, it's not appropriate. So it's not just mitigating the compounds, it's also, or researching the compounds, it's also gaining knowledge of where best to apply this knowledge to. And it can depend on a whole variety of things. This is largely just off the top of my head. Severity of the bloom, location, logistical considerations, cost-benefit analysis. And lately we've been playing with actually combining technologies, not just using one, but possibly two, one to target cells and another to target toxins, possibly. Ideally, it'd be great if we could target blooms offshore before they start. In real life, that's a problem at the moment. These initiating populations, very low concentration, they're at depth over a large area. We can't see them with satellites. In fact, we mostly can't detect them with the instruments that we have, except for a biological or a sample that we put under a microscope. And there's also the ecological question. What is the function of red tides? They've been here forever. The Eastern Gulf of Mexico is incredibly productive, but these waters are oligotrophic. We know that red tides are very productive. They fix a lot of carbon in the system, produce a lot of oxygen. If we're preventing these blooms, are we altering productivity of the Eastern Gulf of Mexico too? These are the research questions we need to look at. But we've basically come to the conclusion we're going to need to target whatever strategy we use to the situation. As I said, an anoxic dead canal or larger bay coastal regions, those are kind of the extreme two ends. Possibly using a combination of strategies, targeting the cells and toxins differently. And lately, we've been also thinking about, well, what if we don't kill the cells? What if we can just slow the growth of Karenia? So hinder it so that the diatoms can outcompete the Karenia. Karenia grows at 0.3 divisions a day. So it takes, for one cell to divide into two, takes about 3 days, versus a diatom grows at 4 divisions a day. So those a diatom cell in those 3 days is going to produce over 1,000 cells. So if we can kind of retard the Karenia so that it gets outcompeted, that's a possibility too. And I just wanted to point out, it's not just enough to deter it or to control the bloom. We need to monitor what we're doing, both to the environment and the bloom itself. So when you see the testing going on, for example, with clay off the dock, yes, we're looking at the effects on cells and toxins, but we're also looking at the effects of that clay as it settles to the bottom. What is it doing to the oysters and clams? What is it doing to the fish in the system? Is it altering the chemistry? And with that, I think I'll conclude and just acknowledge the staff here at the Institute, all our technicians, one of which I just lost actually to you guys in a PhD program. But I won't hold that against the University of Florida.
Sorry about that.
The Economos and Barancik Foundations, as well as the NOAA EcoHab program, the Florida Sea Grant, and the Florida Red Tide Initiative. And I just want to acknowledge a lot of this research I presented is collaborative. We have 5 different red tide groups here at Mote, and we all are working together on the mitigation issues at the moment. So with that, I'd be quite happy to take any questions anybody has. And let me stop the sharing.
Yep. Thanks so much, Dr. Heil. It was a great talk. I mean, no magic bullet or no silver bullet, I think is, something we all need to deal with and just get over. And so I think the great palette that you're developing is supremely interesting and seems like some more promising in some situations than others. So let's open it up to questions in the Zoom room. As you're thinking about your question, I'll read one that came in from YouTube from Dr. Zhang, who's here at UF. Could you discuss how citizen science data like the beach condition reporting system could potentially help in the research on toxic algae? Are such data largely used for monitoring or forecasting blooms in the same way that remote sensing data are being used?
Well, citizen science is very appealing because you can A, involve the public, and B, expand your monitoring capabilities. And in certain cases, extend your research capabilities. I know they're using citizen science with the HAB scope right now, which is a microscope with a video camera attached, which essentially doesn't identify the Karenia microscopically. It identifies it, how it swims, and has an algorithm that pulls it in. If you get enough of those, you can have quite extensive coverage, and that's the basis for forecasting respiratory impacts, which is underway now. And the thing is, you need the high resolution with something like that, because that's very dependent on concentration, which varies with winds and tides, sometimes on an hourly basis, over a large scale. And that's the appeal of citizen science. Right now, we're also developing qPCR for citizen science. There's some new qPCR technology that actually talks to a cell phone. It's a simplified kit with 3 or 4 steps. It could be used by, in our case, a well-educated citizen science, but have that molecular data reported back too. And the key thing here is once a bloom is established, you're non-regulatory. You've already closed all the shellfish beds. And that's important because legally the FDA requires us to use microscopic counts to provide the data to close the shellfish bed. So once you've closed all the beds, then citizen science comes in handy for predicting local impacts. And we're developing tools that incorporate this just to expand the coverage as well as involve the public too.
Yeah, that's great. I think it's a great set of data to add to all of these, prediction or even just, trying to elucidate when, where, why, and you have the coverage to ask that in a much broader way than even the excellent FWC data, which are, validated and under the microscope and everything. But if you begin to see the connections between the two, you can expand your scope.
Well, each one of those data points in an FWR map is probably 2 to 3 hours of effort, and somebody's sitting in a microscope. And exactly. Yeah, my people used to do that, and the amount of effort behind one of those maps is underappreciated.
For sure. We have a question from Hugh. Go ahead, Dr. Fan.
Thank you, David. Thank you, Cynthia, for a nice talk. It's very comprehensive. I learned a lot from your talk. I have 2 questions for you. One is at the beginning of your talk, you talk about that current cause those the basic red tide. My question is basically what's the effect of hurricanes on the generation of red tide? The second question related to the mitigation. You talk about a lot of the chemicals or biological species try to mitigate that red tide. In the one slides, you talk, I'm just curious about physical approach instead of all those chemical approach. But I realized in one of the project founded by the initiative that's called UVC, and then I'm just curious what's basically results from those physical approach.
Oh, the physical approaches, yeah. In terms of hurricanes, that's always been a question. If you look at the past history of hurricanes, sometimes they disrupt a bloom. More often, they'll take it and move it up the coast or actually spread it if they're moving the entire water mass. One of the things we're looking at right now with our large NOAA EcoHab grant is the effects of hurricanes on red tides, because we're concerned with climate change actually increasing the severity of red tide. It's not red tide, hurricanes. We're seeing bigger storms with more rainfall runoff, so over a shorter time period, so more of a nutrient pulse into a red tide. What led us to be interested in this is 2018 was a bad storm year, bad red tide year. 2005 was a very bad red tide year. And that was the year we had 3 back-to-back hurricanes in South Florida. So this is something that interests us greatly and is the topic of ongoing research. Florida has a long-term database that dates from 57. It's problematic it's got over 100,000 georeferenced Karenia counts. It's problematic for a variety of reasons statistically because it's essentially just a bin where all the counts were gathered. It's not one station sampled every week over time, but it is useful for things like looking at the effects of hurricanes over time, which is what we're kind of throwing at it right now. In terms of physical things, there are projects that are looking at this. Bubbling is one thing people are looking at. That's the one that comes to mind right away. Turbulence is very detrimental to Karenia, at least in cultures. So anything that increases turbulence could potentially have a negative impact on blooms. And that's appealing because you're just bubbling, bubbling the water. You're not adding anything to it per se. I'm thinking more in terms of chemical. That's the one that comes to mind about the physical. But if you're seeing gaps there, and I listed some of the projects, I'd encourage you to think about submitting a proposal because they're considering anything that is reasonable and can be looked at on a scientific basis in that time period of the grant. So that's a short answer. We know a lot about the physics of the system in terms of initiating blooms and developing them. The other thing we're looking at in that grant, besides hurricanes, is what factors terminate blooms. And physics is one of the things we're looking at. And it's kind of interesting because we've found in the last 20 years there are certain patterns of bloom termination. And I suspect a lot of those are physically related, but it's more on a larger coastal scale. It's not something that we can actually try to mitigate, at least at the moment. Right. So I wandered a little, but I hope I answered your question.
Thank you.
Thanks. Dr. Clark, please. Go ahead, Mark.
Thank you, Cynthia. Great talk. I really enjoyed how you've kind of laid out the complexity of this testing process and evaluation process. Really interesting to see what you've all got there as kind of the structure. My question relates to, and I thought I saw humic acid as one of your compounds to be tested, and I thought it had green lights, but I didn't hear you talking about it. But I could have been confused based on the way it kind of ranked out, and it seemed like there was a bit of a alignment issue on that table. So I was just curious with regards to humic acids, and it relates a little bit to the idea that most of those humic acids, I'm assuming, are terrestrial based, and so as you have a runoff event, you might get a nutrient pulse. But if there was a reduction at all, I mean, could you also get this kind of reduction pulse at the same time?
That was at the bottom. And we've looked at humics with regards to if they affected the cells and the toxins, and they didn't. But what they do affect is aerosolization. So that little thing at the bottom was a whole separate set of experiments that just looked at aerosolization. We know that they had no effects on the cells themselves, although that was actually my dissertation topic was humics. The cells can take up humic acids as a carbon source and a nitrogen source. So it's a nutrient source, but it doesn't seem to affect the cells and toxins in our 48-hour experiments. So that was primarily looking at the effects upon aerosolization of higher concentrations, probably, I've got to think back, 2 to 5 milligrams per liter, what you typically find in an estuary. When you get the runoff, we do get a lot of humics into our environment. I'm thinking in terms of the Peace River and Charlotte Harbor, it turns essentially brown. When we get a hurricane dump like that, I've been out on ships, and essentially we don't get much growth of anything for about a week. And it's a combination of the humic acids and suspended matter in the system. It just dumps so much suspended matter that you're limiting light for everything, including red tide. But once that settles, we looked at the hurricanes. On red tide and the other things. Once those suspended sediments settle out, you do tend to get a big bloom, but because you've gotten a big pulse in nitrate, it tends to be a diatom bloom in most cases. We're looking at the effects on red tide too. We haven't had a ship fortuitously out there at the right time to look at that, to tell you the truth. But I'm interested in because I'm kind of wandering a little bit around here as I pull my thoughts together on this. You're getting a lot of suspended sediments, you're getting a lot of humics, you're getting primarily nitrate. And nitrate tends to favor diatoms rather than Karenia. Karenia prefers ammonia and dissolved organic nitrogen. That being said, it can take up nitrate. So that's sort of an open question on what the hurricanes are doing to the systems in terms of a lot of humic inputs.
Thank you.
All right, we have a
Although I
question
answered
from,
that
I think we might have time for one more. This is from Christine from the field. So Christine says, I'm intrigued by some of the natural compounds being explored like turmeric. Can these products be sourced at the scale relevant for whole bloom treatment? I mean, I know I take my turmeric every night for my inflammation, so I'll donate mine, but what about, I mean, at the scale of like offshore type of treatment as opposed to canal type?
Well, that's come up, and it turns out one of our postdocs is from Hawaii, and she returned there. There's actually an industry in Hawaii that's growing turmeric, and they have a variety of different strains they're testing for effectiveness. But we've been talking to them about sourcing larger quantities, and yeah, that's an issue with some of the compounds that we've identified in the literature. Either they're extraordinarily expensive for gram quantities, or you have to buy a ton of the stuff. Some strange Chinese herb that worked remarkably well, but you need to buy a ton of it on Alibaba to source it, which is not, yeah, they wouldn't thank me if a ton of this compound showed up on our loading dock.
Yeah.
So yeah, that's one of the logistical considerations that you really have to think about.
All right, well, let's give Dr. Heil another round of applause. Thank you so much, Cindy, for speaking with us today. It was really great.
Oh, thank you.
You know, we've heard a lot of amazing talks this semester, often with a little bit of bad news at their core. And so, you know, to celebrate the progress you made is, you know, to keep us all thinking there is a path forward here, but it's a hard road. And we appreciate that kind of guided tour you gave us. Thank you so much.
And I'm encouraging you, if you have ideas, the request for proposals is coming out in September. And it's not very involved. So I encourage you to apply.
You'll be seeing a bunch of UF proposals. So we appreciate that a lot. All right. Thanks very, very much, everyone. We'll see you next week. Same time, same place. Have a good week. Be good to each other. Be good to yourself. Cheers.
Thank you.